Monday and everything. Okay, so last chapter we talked about the cell cycle. Anybody remember what the first phase of the cell cycle was? Interphase. It was interphase. And what happens in interphase? It had three parts. The first one was called G1. What happens in G1? Things grow, right? And then it was S phase. And S phase stands for synthesis. And synthesis is copying parts. And then it got ready for mitosis. Now mitosis had four phases. Anybody remember what the letters are for mitosis? Remember PMAT? Shake your head, yes? Yeah, PMAT. What's the P stand for? Prophase. In prophase, remember the nucleus disappeared and the chromatin coiled up and made chromosomes. What's the M stand for? Metaphase. What happens in metaphase? Chromosomes line up or they meet in the middle. What's the A phase? Anaphase, and they do what? Yeah, they go away from each other, right? And then we have telophase, where the nucleus starts to reform, the chromosomes start to uncoil, the cell starts to divide. And then what was the last part called? Cytokinesis. Okay, very good. Okay, so Rhea had a parent cell and it went through the cell cycle. What did Rhea end up with? How many cells? 2. What could you tell me about those 2 cells? They're identical to each other and they're identical to the parent. Now, what cells in our body will undergo cell cycle? Just a few of them or most of them? Yeah, most of them, right? But we have a few cells that we do not want to be exact copies. So we are going to talk about those today in meiosis. But we're going to start out with a little vocabulary, of course. And we know that a gamete is a sex cell, and yes, you do need to be filling these in. We have 2 types of sex cells, which makes sense. We have the sperm, which is the male sex cell. And we have what's called the ovum. Now, ovum is the technical name for egg. We tend to just say the egg cell, but ovum is more correct. And the plural of ovum is not ovums, it's ova. So if you see this word and you see this word, they mean basically the same thing. You just have more of the ova than you have of the ovum. Testes. We do not have balls in this class, we have testes in this class. Let's keep it scientific. And it's the structure that produces the sperm. We have the ovary that produces the eggs. The babies do not develop in the ovaries. The only thing the ovaries do, and they're actually fairly small, is they're going to make the ova or the egg cells. And ladies, at this point, you have all of the ova that you're going to have. Your body's not going to make any more. It's going to finish processing them, but they're all there. Which is why when you run out of ova, you go into menopause. Gentlemen, you can make sperm the rest of your life. Unfortunately, the older you get, the more susceptible the sperm is to mutations. We have a zygote. We've talked about zygotes. It's a fertilized egg or a cell that's formed when an egg and sperm cell unite. Fertilization is the joining of the egg cell and the sperm cell. Haploid means one set of chromosomes. And again, set is important right here. We have diploid, which is 2 sets. Again, if you can remember these in a pair, I always know that di means 2. So if I'm diploid, I have 2 sets, and then the other one has to be haploid, and that means it just has half as many. Again, the HA reminding me it's half as many. Who do we get our 2 sets of chromosomes from? Yeah, from our parents. We get one from mom, we get one from dad. We bring them together and we make a gene. Okay, homologs, these are corresponding chromosomes. And we've talked about this a little bit. We said that we get a chromosome from mom for blue eyes, we get a chromosome from dad for brown eyes. Those 2 chromosomes code for the same trait, but they are not identical. So again, they're corresponding chromosomes, coding for the same thing, but not necessarily identical. And a lot of times you'll see this, I'll usually say homologous chromosomes. That's just what I've always called them. But homologs and homologous chromosomes are the same thing. Okay, so enough of review. So meiosis in summary is, we have 2 nuclear divisions, so we have 2 times, the nucleus is going to divide 2 times. And so we go through PMAT twice. And we're just creating haploid gametes. And again, because we're dividing it twice, we only have half as many of our chromosomes. And this is we have a process here that's called crossing over, which we will talk about here in just a second. And again, 4 daughter cells are created. We said the cell cycle, we have mitosis, it's going to make 2. And if you think about it, mitosis has the word 2 in the middle of it. That helps me to remember it's going to make 2 cells. And if you remember that, meiosis is the other one. It's going to make 4 daughter cells. And they are not going to be identical to anything. They're all going to be their own unique character. Okay, and all this is going to lead to genetic recombination. That's why you may be similar to parents, but you're not identical to your parents. Okay, there are technically 11 steps in meiosis. First one we already know. First one's interphase, and it's not even on here, okay? So we haven't quite got to meiosis yet. Interphase, our cell is just growing, doing its thing, starting to copy itself, getting ready to divide. From there we go to meiosis 1, and you see this little Roman numeral 1 right here? That's extremely important when you're talking about meiosis. So, yes, you do need that. Okay, again, prophase, said we was going to go through PMAT. Prophase 1, again, we got that Roman numeral. Don't forget it. Chromosomes condense. Nuclear envelope disappears. Spindles are formed. Again, this is the same thing we saw in the cell cycle. Homologs are going to pair up. So my chromosome from mom, my chromosome from dad are going to find each other. And they're going to pair up. And crossing over occurs here. And I'll talk about that here in just a second. But you can see in the picture up here, you can see how our blue chromosomes have matched up, how our purple chromosomes have matched up. We can see our nuclear envelope is breaking down. Again, prophase 1. Okay, and again, here's a little bit of crossing over. Again, we're going to come back to it here in just a second, like right now. Okay, we said that chromosomes have 2 chromatids. So we made a nice X structure. So here's my chromatid, or my chromosome. Each one of these is a chromatid. Well, when these pair up, they're going to be super, super close together. And what happens, so here's my chromosome, here's my chromosome. They pair up. The legs will get entwined. My chromatids will get entwined. And what happens, they'll break off and then they'll reattach. But when they reattach, they reattach in a different location. This is called non-recombinant. If it stays straight, it's called recombinant. But again, this is the crossing over process. So you can imagine there's genes all along here, there's genes all along here. If we're crisscrossing and matching and crossing over, we're changing the genes from this chromosome over to this chromosome. So again, it's just leading to diversity. Okay, metaphase 1, same old metaphase. My homologous chromosomes are going to move to the equator. So we're going to have pairs of chromosomes because they're homologous. And again, you can see our pairs here. And they are going to go to the equator of my cell. Anaphase, this is the step that is very, very different than the cell cycle. My homologous chromosomes are going to separate. the chromatids do not separate. So the chromosomes are still going to be diploid because each chromosome is still going to have 2 sets of genetic material. So again, you can see here, you can see the entire X getting pulled this way, the entire X getting pulled this way. So again, here's mom's genetic material, here's dad's genetic material. Still have 2 sets, so still diploid. And then telophase 1 is cytokinesis is basically what we had before. Normally the nucleus won't completely reform, but things will kind of regroup into a nuclear type formation. Because we only have half the number of chromosomes, we reduced our chromosome count, but they are still diploid, or diploid, however you will say it. Okay. We do not undergo interphase 2. Anybody want to take a gander on why we do not undergo interphase 2? What happens in interphase that we don't want to happen again?
It grows.
Yeah, it grows. Okay, what else happens in there that we don't want to happen again? What was the middle phase in interphase? Was the S phase, right? What happened in the S phase? They copied everything, right? Well, if you're trying to get rid of half of your chromosomes, you don't want to have to copy everything, because that's just kind of defeating the purpose. We're just going to be in a vicious cycle going round and round and round and round. We don't want to do that. So we don't have an interphase 2. We just go straight to meiosis 2. And again, there's no replication. And it presides just like mitosis, except it starts and ends with haploid cells instead of diploid cells. That's the only difference. So again, if you think back to mitosis, you got this down. Okay, so we started out with a single egg right here. We underwent meiosis 1. We now have 2 cells, and each cell has a fully diploid chromosome. Metaphase 2, those chromosomes are going to meet in the middle. Anaphase 2, my chromosome is going to separate. Chromatids are going to go in each direction. So now these guys are haploid, and they just have 1 set of chromosomes. Telophase 2, nucleus starts to reform, cell starts to divide. Cytokinesis, our cells are divided. Again, we started out with 1. We now have 4. So why is it important to have only half as many cells or chromosomes in our sex cells? Why is it that important? Anybody have a guess? What would happen if they were not half as many? What happens if we get two chromosomes from dad and 2 chromosomes from mom coding for the same thing? What's going to happen?
Mutation.
Could be, yeah, but what kind of mutation? Genetic, okay. How many chromosomes would we have if we put
4.
2 and 2 together? We'd have 4. If we have 4 chromosomes for a head, how many heads would we have? We'd have 2. How many arms would we have? 4. How many legs would we have?
4.
Okay, now that may sound really cool, but then the next generation comes along and I've got 4 chromosomes and 4 chromosomes, and now I make 8. So I have 4 heads and 8 legs and 8 arms, and the next one is going to be, yeah, the next generation is going to be 16, and the generation after that's going to be 32. And after a few generations, we're just going to be weird, okay? So that's why it's super, super important
We're just going to be a ball of arms.
that we get rid of half of our chromosomes, okay? Now, another term that you see a lot of times with this is a term that's called gene linkage. And it's just like it sounds. The genes are linked together. For the most part, when we was doing all our Punnett squares and everything, we was dealing with individual genes. Well, those genes are put together to make a chromosome. So chromosome is just a bunch of linked genes. And what happens is that those genes want to go or to separate together or segregate together. So here's my chromosome. These genes want to stick together. They like each other and they want to stay together unless there's crossing over. They're going to segregate together. And example of this, most blondes have what color hair? Blonde, okay? And the reason that blonde hair and blue eyes go together is because those genes are on the same chromosome and they're linked. And unless you have crossing over, which you do have crossing over because you'll see blonde people with brown eyes, but if you have blonde hair and blue eyes that are on the same chromosome and they just like each other and they want to stay together. Again, that's just gene linkage. And again, the only thing that's going to separate this is that crossing over. And it's amazing the diversity we get because of crossing over. This is not something that just happens occasionally. This happens almost every time your homologous chromosomes come together. And again, just comparing cell cycle or mitosis with meiosis, we start out with a single cell that's undergoing interphase. In cell cycle, we come straight over here, go through our PMAT. We end up with two cells that were identical to our first cell. If we go into meiosis, we go through meiosis 1, makes two cells that have half as many chromosomes. And then we go through metaphase 2, where those chromosomes are going to split the chromatids, and we end up with 4 daughter cells. So we've got a couple of questions right here. You guys answer them on your Chromebooks right now. Normal body cells have how many sets of chromosomes? 2. What is the word that means 2 sets of chromosomes? Diploid or diploid. Sex cells have how many sets of chromosomes? 1. And what's the term? Haploid. Okay. Turn your notes in. Come on.